Hi everyone, welcome back to my channel. A little disclaimer before we begin. I have really bad cold and cough right now, so please bear with my voice throughout this video. Yeah. Coming back to welcoming you all. So the today's video is on nuclear run-on transcription, which comes from a molecular biology. And this technique is basically used to study the gene expression and regulation. If I have to make you understand within a line or two, I would say it's a laboratory technique which is used to study which genes are actively being transcribed in a cell at what rate, and it provides you a snapshot of the genes that are currently being expressed. So with this, let's move on. You will ask me that why are we focusing today on nuclear run-on transcription if we have other methods such as primer extension, there is S1 mapping, and also northern blotting. But let me tell you, this is different from other method because the other method will only give you the information about the concentration of specific transcript in a cell at a given time. Okay, they will not necessarily tell you about the rate of synthesis of the transcripts in vivo. And that's how this method comes into picture because it will let us know about the rate of synthesis, particularly of the transcript in vivo. Okay, so and you will ask me then why the other methods are not giving us this result, because the transcript concentration depends not only on its rate of synthesis but also on its rate of degradation. Okay, so to measure transcription rates we can also employ other methods including the nuclear run-on transcription, and there is one more method which is known as reporter gene expression method. Let's move on. Before getting into the process and the significance, let's refresh one's memory because it's very important in order to avoid any further confusions by the terminology. So for those who don't know what is transcription, transcription is basically a process wherein there is a synthesization of a single stranded mRNA or messenger RNA molecule from a double stranded DNA molecule, and this process is simply termed as transcription. Okay, then most of the time people get confused between these two terminology, that is in vivo and in vitro. So it's very simple to understand. In vivo is something that is done within or with uh, entire living organism, and in vitro is something done outside the living organism. That is very simple definition. And then the last one is the definition of rate of transcription. Now you know what is transcription, so then you will definitely understand what is rate of transcription. Rate of transcription is nothing but the rate of production, that at what speed the mRNA is getting synthesized from the DNA molecule, and that is rate of transcription. That's all. Now let's see what is nuclear run-on transcription. Okay. Now, if I have to tell you what is nuclear run-on transcription, I will explain you the process, and that's how you will understand what this method is all about. Okay, what is the idea of this method? Now here what you do is you basically isolate a nuclei from the cell. Consider this is the cell, okay, blue one. This red color is the nuclei, and you have isolated this nuclei. Okay, now you have to focus that you have to isolate the nuclei from the cell, and then you have to allow them to extend in vitro the transcripts that had already started in vivo. Okay, and this continuation of the transcription in the isolated nuclei is called run-on transcription. Let me explain you very simply. Just assume that you are a researcher. You have one cell In cell you have one nuclei, okay, and in that particular nuclei there is transcription begun at that particular stage, okay, of some gene. I'm not naming the gene, just assume there's one gene whose transcription has already begun with the help of RNA polymerase, okay, which means that there is some strand which has formed of mRNA, okay. Now at that moment you have isolated the nuclei. Now there is a shift. When the nuclei was inside the cell, it was in vivo situation. And now the nuclei is outside the cell, and whatever you're going to do is outside the cell, which means the condition is in vitro. Okay. Now in vitro, what you have to do is you have to allow that transcription to happen. Now what will happen? The initiation was already begun when the nuclei was inside the cell. And now in the next step, you're allowing the elongation of that strand with the help of RNA polymerase in vitro situation. Cool. Now how we are going to do that? And if you have already allowed, how will you find out that which strand was developed in vivo, which strand was developed in vitro? So since we know that we have already performing everything now in vitro, we will use any labeled nucleotide. So with that help, you can find out, okay, it will help, it will act as a marker for you to find out. I hope this is very easy to understand. Okay. So where was I? I was saying you that you have to allow them to extend in vitro transcript that had already started in vivo. Okay. And this continued transcription in the isolated nuclei is called run-on transcription because the RNA polymerase, which has already initiated the transcription in vivo, simply runs on or it continues to elongate the same RNA chain. Okay. And as I already told you that the run-on reaction is usually done in the presence of the labeled nucleotide. So the products will be labeled. And because the initiation of the new RNA chain in isolated nuclei does not generally occur, so one can be fairly confident that transcription observed in the isolated nuclei is simply a continuation of the transcription that was already occurring in vivo. Okay. And therefore the transcripts obtained in the run-on reaction should reveal not only the transcription rates, but it will also give us an idea about which genes are transcribed in vivo. So to eliminate any possibility, okay, of initiation of a new RNA chains in vitro, which does not happen usually, you can still add an anionic polysaccharide, for example, heparin, which will bind to any RNA polymerase if present, and it will prevent the reinitiation. Okay. Now, ये सब तो हो गया. What will you do later? How will you find out? So once the labeled run-on transcripts have been produced, they must be identified. Cool. So the easiest way to perform the identification is by the dot blotting. I'm so sorry. I'm so sorry. So the dot blotting method, okay. So the samples of the known DNA, known denatured DNA are spotted on a filter, and this dot blot is hybridized to labeled run-on RNA. The RNA will be identified by the DNA to which it eventually hybridizes. यहाँ पे dark color का जो दिख रहा है that signifies hybridization. Okay. So furthermore, the relative activity of the given gene is proportional to the degree of hybridization to the DNA from that gene. इसको basically conclude ऐसे करते हो जितना ज़्यादा dark दिखेगा उतना ज़्यादा hybridization हुआ है. Okay. This is what I mean when I say the degree of hybridization. Okay. So not only this, but the conditions in the run-on reaction can also be manipulated and the effects on the products can be measured. For example, inhibitors of certain RNA polymerases, for example, heparin हम डाल देंगे. Okay. So that time, are, for example, the inhibitors जैसे मैंने कहा, if you add that, so uh, you can check whether it's inhibiting the transcription of any particular gene or not, and if so, the RNA polymerase responsible for transcribing that gene can be identified. Okay, now the same thing is explained in a pictorial clear representation. ठीक है, I've taken this image from the reference book directly. तो मैं यहाँ पे I will not take much of your time. It is self explanatory now. मैं एकदम simply इसको explain करती हूँ. This is the cell. Okay, now this is the nuclei, this yellow color का. You take the nuclei out. Okay, this is the DNA molecule. These are the gene X, Y, and Z gene. Here, this orange color is the RNA polymerase. I don't know whether you can see there's this dark blue line. Okay, this is where the RNA polymerase has started the transcription of the only Y gene. Okay, so Y gene का transcription it has already started, और तभी हमने इसको isolate कर लिया nuclei को. So now what you will do is you will incubate with the labeled nucleotide, और वो label nucleotide के साथ you will let it elongate. So once the elongation takes place, this will extract the RNA and then use the dot blot assay, और यहाँ पे XYZ gene के DNAs को रखना है on or nitrocellulose or any other suitable medium. Okay, and let the now here what do you see that or any other medium you can use. Okay, it could be nitrocellulose or any other thing. So and hybridize the blot to the labeled run on transcript. Now because the gene Y was transcribed in the run on reaction, its transcript will be labeled and the gene Y spot becomes labeled. Can you see here that this dark color ka jo hai wo? Okay, cool. So the more active the transcription of gene Y, the more intense the labeling will be. On the other hand, if you see the X and Z genes, they were not active, so they are no labeled. Okay, so there are no labeled X and Z transcripts are made, and that's why the spot X and spot for the Z gene remains unlabeled. I hope I made it clear. This is just a procedure, so I will not take you through. Significance, I've already told you. हम effects check कर सकते हैं of the enzyme or degree of the activation of the gene, and there are many more other significances as well. So this is the reference. This is the book called Molecular Biology by Robert Weaver. It's a fifth edition, chapter 5. I've also told you the page number. You can go back, look at it, and try to understand more. With this, I thank you so much. I know it's a tough topic to understand, but I've tried my best to make it simpler and make you understand. But if you watch this video like twice or thrice, it will be clear. And for your reference, I've told you the book's name as well. You can go back and read. And still, if you think if you require any help, you can post down in the comment. And for my own motivation, if you've liked and you think this video is helpful, so please do like, share, and comment on this video, and do subscribe to my channel. And I'm really so sorry for my bad voice, and I was continuously sneezing and coughing because I have severe cold and cough right now, but still I wanted to make this video, so did I. And yeah, with this, thank you so much. Please take care of yourself. Till we meet next time.